Hello, welcome to our LibreHub seminar series. Today, I'm thrilled to present Dr. Weijian Zong. So, Dr. Zong holds a degree in electrical engineering from Peking University. He did his PhD obtained in 2017, where he focused on developing imaging technologies for neuroscience. He's currently a researcher in the Moser Group at Kavli Institute for Systems Neuroscience. In the past few years, he led the development of free versions of miniature two-photon microscopes to record neuronal activity in freely moving rodents. Besides the miniature 2P microscopes, he also developed a series of other advanced fluorescence imaging tools across broad topics. His current research is developing all-optical approaches for characterizing functional connectivity in the medial entorhinal cortex. Dr. Song also holds 6 invention patents in optical imaging. His work on developing a new generation of miniature 2P microscopes was selected as China's top 10 scientific achievements in 2017. He has received awards such as the Shizan Pai Young Biophysicist Award in 2017 and became a Marie Sodowska-Curie Fellow in 2019. So I'm very pleased that we have the opportunity to host his talk in this seminar, where he will talk about this open source miniature two-photon microscope for large scale calcium imaging in freely moving mice. So Weijian, please go ahead and share your screen, and we can start with your talk. This talk will be posted online as it will be recorded.
Okay.
We can see your PowerPoint screen now.
Yeah. Can you hear me?
Yes, you can hear you well.
Yeah.
And now see the slide correct. Excellent. Go ahead.
Thank you for the introduction, and so sorry for this mistake of the time zone problem, but I hope that the people who are interested can see the presentation later. And so it's my great honor to present our recent work on the development of this miniature microscope that can be used in the free moving mouse. So I will start by introducing the purpose why we want to make such thing small. So I think one of the major goal of the 21st century neuroscience is to understand the neuron substrate of behaviors. And most of the neuroscientists now have agreed that the behaviors, maybe I should have a pointer somewhere.
We can see your mouse pointer.
Oh, you can. Okay, great. Yeah. All right. Yeah. You can see the mouse here, right?
Yes.
Perfect. Yeah. Okay. So the complex behavior of mammalians, such as the mating, fighting, our social interaction, were generated by the functional brain, which automatically was produced by the activity of the millions of neurons. So therefore, in order to understand the substrate of neuron substrate of the behaviors, we need to record large-scale neuron activities during behaviors, and then to identify the functional cell types, which means how these neurons are firing or tuning to specific behaviors, and then we can unveil their contribution to this cognitive function, including behaviors. So, therefore, the scientists have looked for the method that record large population of neural activities in rodent brain for decades of years. And one attractive way to do this large population recording is a calcium imaging. So the action potential of the neurons can introduce the increasing of the calcium concentration in the cell body. And this increasing of calcium concentration, which is called calcium transit, can then be reported by the sensitive calcium indicator, such as the famous GCAMP family. And then the fluctuation of the signal from this calcium indicator can then be recorded by the state-of-the-art microscope, such as the multi-photon microscopes. And you can see that the resolution of the multi-photon microscope is really high because it really limited the background signals from the out of focus plane. So you can see this is the one photon focus that you see a lot of signal on the background, and this is the two photon focus that there's no signals from the out of focus plane, which give you really high spatial resolution and good optical sectioning capabilities. And then the cell yield of this method can be over thousands of cells with a spatial resolution that is less than 1 micrometers. So however, one biggest limitation of the traditional multi-photon microscope is that the system is really complex and huge. So the animals had to be fixed under the microscope when the recording is ongoing. And to mimic the real environment that the animals can experience, the researchers, they usually built up this virtual reality surround these systems. So in this way that they can mimic the environment that the animals experience every day. However, the people have found that actually, even you can build up an virtual reality, the neural activities in the real world are quite different from the same neuron that is in this virtual reality. You can see these two examples, which are the place cells in hippocampus, which has a very clearly firing field in the real world, but their firing much more messy in the virtual reality. And which we believe is because when you had fixed animals, you actually removed a lot of central input, such as this vertebrate feedbacks from the air or all of this viscous touching. And your head direction systems is also not working because you had fixed animals. The animals cannot freely move its head. So this network doesn't give any input to the place, to the space representation. And all of that make this firing in the virtual reality totally different from the real world. So therefore, the scientists start to develop the miniature microscope that can be mounted at head, so the animals can freely move in the environment when we recorded the activities. And the story started at 2001. The first prototype was published from the Benford Denk groups, which is the pioneers of two-photon microscope. But at the beginning, this miniature microscope is still quite heavy that cannot be used in the small rodents such as mouse. And the several works that are that were followed up also develop different parameters, but still that none of them are really working in the free moving animals. And the problem of this earlier age miniature microscope are, first of all, that the dispersion in this excitation fiber, that the decrease the excitation efficiency. So you cannot have a really high power pose that can be used to do the multi-photon imaging. Also, the small 2D scanner, which is necessary to do this raster scanning in the two-photon microscope, were quite slow, and scanning angle were also very small at that age. So you cannot have a really big field of view to image many neurons. And moreover, there's no suitable miniature Z scanning devices that can be integrated into a Miniscope to change the focal plane. And because of the miniature optical and electrical devices development really fast in these recent years, you see an emerging new technology that are published of these miniature devices. And I do not have time to go through all of these beautiful works, but only mention two of the works that I was involved when I was in Beijing. So in 2017, we published of the new our first version of this miniature two-photon microscope. And in this microscope, we developed a new type of the hollow curve photonic crystal fiber that can deliver the sub 100 femtosecond laser in 920 nanometer, which is the best wavelength to exact the GCAM indicators. And you can see that we can transfer more than hundreds of milliwatts of the laser without expanded this pulse. You can see that the pulse still kept about 100 femtoseconds after 1 meter of this fiber. And with this new fiber, and we can have really high resolution imaging in the Miniscope. You can see that single cell can be well distinct from each other. So, however, one biggest problem of this first version of a Miniscope is the small field of view. And you can see that this is the maximum field of view we can achieve with this Miniscope, that only a few neurons can be detected in with this microscope. And also, we didn't achieve any Z-scanning functions in this Miniscope. So we can only mount the scope in one certain plane, and then that's it. We cannot change the focal plane. So in order to solve these two problems, we published our second version of a Miniscope in 2021. And this time, we expanded our field of view to more than 400 microns. So hundreds of neurons can be recorded from single planes. And we also produced the prototype of the scanning module in this Miniscope by using a commercialized, smallest commercialized available electricity tunable lens that can change the focal plane when you give different current. And with this small Z-scanning module, we can change the focal plane in a range of 180 microns. Again, you can see that the neurons and different layers can be detected with this Miniscope. However, the problem is that in order to increase in the field of view and introduce this scanning module, the weight of this Miniscope is much heavier than the first version, which reached to 4.2 grams. And also, this scope had to using a very heavy and stiff detection fiber to collect enough photons, which restrict animals' behaviors. And another problem is that this electricity tunable lens we are using, they have a really severe thermal effect, which means that when we drive this electricity tunable lens to its maximum output power, we have to use 200 milliamp, and that increasing the temperature about 25 degrees only after about 5 or 10 minutes. And this increasing of temperature will drift your focal plane, which means that you can never track the same cell for more than 10 minutes. So, and all of these problems motivate us to develop a new version of the miniature microscope, which was published this year, and we call it Mini2P. And in this time, we focus on increasing cell, not only on increasing the cell yield, but also to improve the improved animals' behavior, so we can really using it in the free-moving animals to do real neuroscience studies. So first of all, we want to explore what is the impact of what's the real impact of the scope weight and the stiffness or thickness of fiber to the animal behaviors. So in order to do that, we do a pilot experiment by using a group of animals that was mounted in a 5-gram dummy scope with the same weight as our previous version of Miniscope, with a 1.5 millimeter thick cable, which is also the same as we use in the version 2 of the Miniscope we published before. And also with a group of animals that we mounted a 3-gram dummy scope and a 0.7 millimeter thick cable, which we believe are producible from the engineering perspective. And also with a control group that is without any scope of fiber. So from this result, you can see that the animals which was mounted 3-gram miniscope and a 0.7 millimeter SIG cables have quite similar exploration ratio compared to control groups. So this is an 80 centimeter box, and all of these gray lines that the trajectory of animals with a 30 minutes exploration experiments. And the animals with the 5-gram weight miniscope and a SIG cable, which is 1.5 millimeter diameter, have much lower expert ratio, which shows that these kinds of weight and of the scope and cable indeed impact animals' behaviors. And you can see that that the total running distance and the mean speed of the animals are all quite similar when animals was mounting a 3-gram miniscope and a SIG cable compared to control group, but it was much worse with a 5-gram scope and the SIG cable. And also the animals, they spend less time in the center of the box when they were mounting a 5-gram scope and the SIG cable, which how they are much less engaged in these foraging tasks. And now we also see that this behavior is not an artifact due to the age or the weight or the of the animals, because that both the age range from 25 gram to 31 grams are involved in these tasks, and you see that it's no strong relationship between the weight and the task performance. And also we included the females and male in the experiment to show that this is also not a fact of the sex of the animals. Then we know that if we can decrease the weight of the scope to 3 grams, and if we can decrease in the diameter of the cable to 0.7 millimeter, we should ensure the most natural behavior of animals, at least in these open field tasks. Then we go back to this second version and found that actually the mean weight, when additional weight to this miniscope came from this scanning module. So in order to decrease the weight of the microscope, we need to change this electric tunable lens to a smaller devices. Therefore, we collaborate with this company in Norway to invent a new version of the miniature tunable lens, which we called micro T lens. This micro T lens using a new technology called the MAPS piezo, so to drive to change the curvature of one lens, and this change of curvature can converge the light and change the final focal plane of the microscopes. And the single micro T lens only weighted 15 milligrams, and in order to increasing the tunable range of the focal focusing range, we stacked 4 T lens together to make this collect micro T lens. And this collect micro T lens also only weighted 60 milligrams, which gave you a focal range of 240 microns with a response time which is only less than 0.3 milliseconds. And on the other hand, we also replaced the materials from the aluminum to an industrial level plastic, which had a similar accuracy like aluminum, which called PEK. And with these two strategies, we're decreasing the weight of the miniscope to 2.4 grams. And then, as in the second step, we want to decrease the weight, the diameter of the fiber. And first of all, we just try to using a smaller fiber bundle, which is the same one we use in this pilot experiment, which was eventually a failure, because you can see that the collection efficiency was also decreased compared to this SIG fiber bundle. And you can see this is the signal from a uniform fluorescent samples. To lift up this collection efficiency, then we added we have an invitation to add a tapered fiberglass on the end of this thin fiber, which have a bigger end of 1.5 millimeter. And then a small n, which is just the same size as this 0.7 millimeter fiber. So the fluorescent photons was collected from this big end of the glass, and after several total internal reflection was collected through this small end to this fiber bundle. Because this tapered fiberglass has a focusing effect, it could bring up the collection efficiency of this tapered fiber bundle to the similar level to the 1.5 millimeter fiber bundle, which is much higher than a pure 0.7 millimeter fiber bundle. And then now we can using this thinner fiber to do the two-photon imaging in the Miniscope. And then I have to go back to the previous problem of the heating effect. And you can see that because it's new tunable lens, they are not using current, but using the static voltage to change the focus. It doesn't have any thermal effect. And you can see that the temperature keep quite stable after almost 1 hour recordings with this micro T lens with a quite stable focus. Therefore, using these new focusing devices, we got quite stable focusing in a long-term recording. And you can see that we can now simultaneously record full plane of neural activity over 30 minutes in free animals. And the neural activity from single plane are quite separatable, which means that we have a really good resolution as well as quite stable focusing capabilities. And with this fast Z-scanning module, we can also do the real 3D functional imaging in the free moving animals. And from this video, you can see that the neural activity from different neurons can be well separatable in different layers of the brain. And the position of the neuron are quite stable, which show the stability of our Z-scanning functions. And in order to use in different brain regions, because of the geometry of brains, we have to use in different optical accession methods. And in this way, we developed 3 different kinds of objectives that can be used to go through a cover glass, which can be used in the cortex that on the top or superficial of the brain, such as the visual cortex, or sensory cortex, or motor cortex, or another objective that can be used to combine with a green lens, which can be used in the deeper brain tissues, such as the hippocampus. And a third objective that can be used to image through a microprism. Then the Miniscope can be used in the cortex that is located on the side view, on the side of the brain, such as PFC or medial and the cortex. And these 3 objectives are interswitchable, which means that we can use the same Miniscope to image different brain cortex and brain regions by just plug in and plug out different objectives. And to show the power of these different objectives, we ourselves successfully record more than hundreds of cells in visual cortex by using just a cross window on the top of the brain, and 100 of cells from the middle and the henna cortex, which is located on the side of the brain. So we have to use a prism to access, or in the hippocampus, which is in the deeper brain region. So we have to using a green lens to relay. And you can see that compared to the miniature 1-photon microscopes or miniature 2-photon microscope, and get quite higher resolution and the signal to not ratio in the exactly same region and with the same group of cells. And we can also track the same cells in a long-term recording. And in this example, in the medial and the cortex, we tracked about 50% of cells, and here it's more than 59% of the same cells over 1 week. And in these recordings, we continuously record the same cortex in over 1 month, and about 36% of cells can be refound in all these 4 recordings. And we also confirmed that only the anatomical shape of this register cells, but also their function are quite stable. You can see that this is the same cells recorded in 4 days across 1 month, and you can see their spatial tuning are quite stable, which confirm that this is exactly the same cells. And we, I mentioned, we want to using this method in different cortex, and with this diverse brain cortex, one of the most difficult region is the MEC, and because of the location of the MEC is in the side of the brain, just in front of the cerebellum, we have to use a prism to access these regions. And now with our new Miniscope, we're able to record more than 900 micron field of view by stitched 4 recordings together. And we confirm that our imaging is located on the medial and dorsal part of MEC by injecting a virus in the C3, retrograded virus in the C3 and DG that only project back to the MEC, but not the surround paracebaculum regions. And you can see that now we are able to see very, very beautiful grid cells, which was found in MEC, which electrophysiology recorded before. And you can see this beautiful grid cell pattern, which just emerged after a few minutes of recordings. And if you remember that at the beginning, when people discovered grid cells with tetrode, every time they can only record a few grid cells. And now with our new miniature 2-photon microscope, we are able to record more than 300, in this example, more than 300 grid cells, which are within 1 animals. And with this grid cell, we also recorded more than 100 border cells and also hundreds of headed red cells. And one striking thing you can see is that all of these grid cells, they are highly confined in the MEC region, but the other headed direction cell, other cell types such as border cells and direction cells, they are located separately from these grid cells and in the surrounding paracebaculum. And if you overlap all of this spatial tuning cell together, you can see this clearly topographic distribution that the grid cells, they are close to each other and in a cluster in MEC, and all the other cell types like border cells, direct cells, and some egocentric cells, they are mixed in other places. And we also published a paper that's specifically focusing on study this function network topographic, and this was published at the same time as these technical papers. And if you are interested in this, in how we quantify this organization of different cell types, you can check this paper. And we are now also able to using this Miniscope to record very highly dense cells in MEC, in hippocampus. And you can see that in the 2 examples, there are almost 1,000 cells can be recorded in a single field of view. And you can see how dense these cells were labeled and recorded. That I think that you almost record every cell in this field of views. And this is one of my favorite video. I really want to show you that in this task, we train the animals to climb and towel to get a reward, and the animals have to be voluntarily jump down from these towers to get another reward. And the thing I want you to focus on is that even in this climbing or jumping or all of these vigorous behaviors, and our recording can still keep quite stable. And you can see, you can focus on this. This is a raw video without any motion correction. And when the animals, after he eat up all the sugar or the reward, he will jump down from this platform. You can see now that even in the jumping, we can still see very, very clearly signals from single neurons, which shows the stability and the performance of our Miniscope, and which tell you that we now can do a lot of behavior studies with our technique. And we quantified these behaviors that, so the animals, even no matter if they are with the Miniscope mounted or without the Miniscope, they have quite a similar climbing speed, and they have quite a similar evolution of the time they need to climb up this tower, as well as the interval between each climb and jump trials, which means that our scope didn't have any impact on animal behaviors. And we also see that the image stability is quite high. And even in these jumping steps, that the motions between adjacent frame is less than 10 micron, which is even better than had fixed animals. And now we think that we not only want to build up these systems in our lab, we also want to spread it into the world. So we actually spent 1 more year to just decreasing the size and the compatibility of our systems. So you can see this is the prototype in the first version of our Mini2P, and now this is the final version that everything, including the computer and the laser driver, can be integrated into this small card, and this whole system can be even moved to different rooms. And so you can use in one system to do different tasks and to be used in different researches. And now we also decrease the complexity to make one Miniscope, and in the total time to be that is needed to build one Miniscope is only 2 days. Actually, most of the time you are just waiting for the glue to dry. So the actual working time is less than 2 hours. So in principle, that you can build a Miniscope in the labs like a factory. And also all of the component that is in the system is modularized, which means that you can build up the one module. And if you want to modify your head fixation or the detection module, you just need to remove this module and make a new module and plug it in. So this really make the system very flexible and can be customized in different researches. And also we do not want the control really complex, and it can be only used if you have some commercialized software. So we try to using the open source, or open available software to control our system. So now our system can be fully controlled without any customized functions by using the scan image, which you can just download from the website. And we also provided multiple tutorials in the YouTube. So you can follow this YouTube to learn how to make this microscope yourselves. But of course, I wouldn't say that everybody can successfully make one Miniscope that is working perfectly. At least you need to have some basic knowledge in optics and electricals. But you can just also download all the materials, because every materials, for example, like the 3D models, all these assembling protocols, also the control software and operation manuals are on the website. So everything is open. And moreover, we also realize that even you open everything, not every lab that can make sure they build up the functional systems. So we decided to regularly organize a workshop, so to invite people to come to our lab to learn how to build and use these systems. So the first workshop will be organized in this December, and and the next year we will have maybe 2 workshops in half 1 year. But we just want to make sure that the lab who already bought the materials can build up the system themselves. So in a summary, we we developed a lightweight two-photon microscope that can be used in the freely moving animals for calcium imaging. So we're now able to record thousands of cells in the free moving animals, and we can record from diverse brain structures. And we ourselves shows the recording of hundreds of cells in visual cortex, in medium and sentinel cortex and hippocampus. And the Mini2P is totally open source, compact, and easy to build. I mean, I think it's easy to build means at least you have some basic optical and electrical knowledge. And we can use this Miniscope not only in the very simple open field tasks, but also in a wide spectrum of behaviors, such as climbing the tower or the escaping behavior I didn't show here, but we indeed had a project working on that. So which means that this opened many possibilities for the behavioral neuroscience. And finally, I will thank the people who are involved in this project. First, my advisor in my postdoc career, and also all the colleagues that are involved in the microscope development and in the different biology project who helped me to make this proof of in principle experiment happen. And also, I would thank the Marie Curie Foundation who support my postdoc research, and also the Kavli Foundation, ERC, and the Norwegian Research Council who support my current research. And I think that's everything I want to show, and it's now open for questions.
Thank you, Weijian, for that tour on this fantastic journey of the portable 2P microscopes. I think with the weight study, you really highlight how important this is to have a very small, lightweight device, right? Yeah. So I wanted to, I have several questions, but I wanted to start first with a very basic, just for orientation, in case anyone who watches, have a question. But so here, if I understand correctly, you have these small electronic devices that do the scanning of the focused light in 3 dimensions, correct?
Yes.
And then the detector is it's a photo detector that integrates all the fluorescence coming out, correct?
Correct, yeah.
Good, very good. And
so this in principle is a raster scanning, it's like a confocal, or the correct. Most traditional two-photon microscope. Yeah.
Excellent. And so I also wanted to highlight another basic point. You showed one slide where you compare with the one photo. So
yeah,
could you just very briefly remind us what's the most important difference of why the one photon Miniscope versus the two-photon Miniscope?
Right. So I mean,
how do you get so much better resolution, right?
Exactly. Yeah, thanks for the questions. I think that fundamentally it's physics, right? So if I go through, if you can still remember these pictures, and I think that's the benefit of multi-photon microscope, because of this two-photon absorption effect, which means that the fluorescence, the fluoropher, they have to absorb the two-photon at the same time to generate the fluorescence, which is really a low chance. And you need really high power to make this two-photon absorption happen. You have to really focus your light into a spot, and also you need to use the femtosecond laser, which have really high peak power. And even with these two strategies, that you can only get high signals in the focus, which means that all the place that this when all of the out of focus place, they will not have the fluorescent emission, and this really confine your signal only came from the focal plane. And in contrast, if you're using one photon imaging and all of this background, because all of this background, they have fluorescence in the brain, and that will emit photons. And all of this out of focus emission will just increase in the background noise, which is exactly what you see in these images, that when you use the one-photon miniscope, which is still quite popular now because it's so simple. It just needs an LED in the miniscope, and it just uses a small camera to detect the signals. But you can see that it's exactly the same field of view, but you have really, really strong background, which decreasing the capability to extract a signal from single cells. But if it's two-photon, all the signals you see, they are from one of the section. And that is the biggest difference. And that really gave you a high resolution.
Good. So I think probably a question that many people will have. So as you said, there are many users of one-photon microscopes. So in fact, we have one, sorry, Miniscope. We have one question here from Isabel, who is in the audience, but she is saying that she presently uses the one-photon Miniscope, and so her question is, what will be the approximate cost to build one mini 2P Miniscope? And I guess the most important cost is probably the two-photon light source, right? But besides that, what are the costs involved in the device that you use?
Yeah, correct. Yeah, this is the questions I think I got every day from email. Well, first of all, I think the cost must be much higher than the open source version of mini one-photon, which say that the UCLA version or other like open source version. And you are right, that's the main core, the majority of the cost comes from the laser. And I think the laser now we are using this I don't know if we're using this fiber laser, which is already much cheaper than the tight fire laser that is commonly used in the two-photon microscope, but still maybe cost, I mean, maybe $60,000, something like that. And without that, I think the other components, like, I mean, all of the components, they are just some glass, some prisms, and that can be like $10,000 or $20,000. So that's the system cost, right? It is higher. But so if you already have a two-photon laser, then you can cut off a lot of costs. And for one miniscope, I think the cost could be, I mean, depends on how many miniscope you want to make, because the objective that costs most in the systems, and the one objective probably cost maybe $3,000 or $4,000. But the beauty is that this objective can be reused, just like in confocal or two-photon microscope. So in principle, you could buy one objective, and then you use in different researches. And without the microscope, the microscope itself could be like maybe less than $1,000 or $2,000, and that's quite affordable for most of the neuroscience labs. And also the point that the miniscope, they can be miniscopes, they can be reused, which means that you probably only need to build one or two miniscopes in the lab. That is different from the electroscopy probes like a tetrode or neuropixels. But I mean but it's still like you can just the comparison yourself to the mini one-photon.
Yeah, so very interesting. All this hardware. So Pierre, did you have any question?
Yes. So, well, first of all, thank you, Dr. Zong, for your talk. It was really interesting. I would like to know about the motivation that why you decide that this technology to be open source or using open source principle, because it's common to see like open source standard bio instruments, but in this case, this one is high specialized instrument. So which was the motivation that this technology to be open source?
Yeah. Thanks for the question. Well, they have a lot of good reason to be on open source, right? I think that's a trait in neuroscience. First of all, we want to spread this technique. And since we do not plan to have a company to sell this component ourselves, of course, I mean they may have the company exist. They build up the commercialized version of this Mini 2P. I mean, I know them both in China. They also have a company. So if you really want to buy a system from company we do not want to do ourselves, you can. But we realized that I mean, before us tools become standardized, like a confocal or whatever, you need to have more validation. So you need people to build up and use it in their research and report the usable the perspective of this system. Then people can believe that, I mean, they want to use it. And that's our motivation. So we want to open source, and then people, the neuroscience labs, they can start to build themselves and use it in their research, and they can to support this technique. And so that's the first motivation. The second motivation is that we also want to involve more people to develop these systems. Because anyway, finally, this system, as we say, is a specialized systems for neuroscience. And it's even more specialized if you consider that everybody, they will have different projects. They may want to image different cortex. They may want to do different tasks. So they may modify these systems. And if you just give a commercialized system and they do not dare to do any modification, then it's useless for their research. Which means that we want people to know how to build up this system themselves, so they can modify it. They can increase in diversity, and they can also further develop the system, because this system is not it's not to the end yet. We still want to increase in the field of view. We still want to do a deeper imaging. We still want to have a lot of features. So we want to expand the developer of these systems. That's why we want to open source
it. Those are very good points. So I had further questions around the same, but so do you expect, so I'm thinking some neuroscience lab, maybe one that uses 1P Miniscopes already, right? They have the behavioral task and so on. They want to use now the 2P Miniscope. So do you expect most of those users, do you think they will have maybe an engineer who builds the 2P Miniscope, or do you think it's more common they will buy one from a company that provides all the parts, for example, even if they are open source?
Yeah, well, I mean, first of all, I mean, I would say that you of course need a motivation to move to 2P, and the motivation, of course, is that they give you a higher resolution. You can have the 3D scanning capability, so you can distinct the cell in different layers. But the one for them, they still work well in some cortex, like the hippocampus, because the hippocampus is very thin. Or if you have some sparse labeling strategy to decrease the background signals. But after all that, if you really want to use 2P, yes, I mean, so currently, I think you have to probably learn how to build yourself, either from us, you can communicate with us, either apply for our workshop, or just learn from the YouTube and the papers. But in the future, we have some plans. So we are trying to build up an image facility, core facility. And actually, we are recruiting for the application scientist who will learn first how to build up the systems, and then he will help the users in the world. He will just train the people in the world how to build up and using the systems, and he may also fly to the labs to help them to build up these systems. So that's in our plan. But currently, probably the best way is to just try to learn how to make it yourself. But also, of course, companies, many companies are also interested in in building these systems. Even we ourselves are not interested, but we still think maybe that to have some company to can provide some service on building up is still valuable. And you can see that I know that like now this one company, they already starting to sell the package of these Miniscopes. So they didn't provide the building up and the testing, but they already starting to sell the packages, and which is called a lab maker. You can check that. Yeah.
Very nice. So I have a couple more questions, but first let me ask if anyone in the audience has any question about the scope. They might be interested.
I have one. In this case, which are the following steps for the Miniscope in terms of hardware? So what do you expect for the next years? What do you think you would improve? I don't know, for example, using for other models maybe, or improving the user interface to get more data.
Yeah. So I think, thanks for the question. From the application perspective, we want more people to use it. We already built up a collaboration with 5 labs. I think 1 in American and 4 in Europe, and they agreed to be the validation site, which means that we will help them to build up these systems, and they will use this system in different actually animal models. And I know that one of them are using this in bird. So we hope to help them to establish this method in different models. And we ourselves will also using this method to image the different cortex in the brain. And we now have, I think, 5, 6 projects that are targeting on different cortex, including MEC, hippocampus, and visual cortex. And from the development perspective, that I will continue to develop it myself. That's my major interest, research interest, and we hope to further increase in the field of view, because current field of view is 500 times 500 microns, and you can get around to the best 1,000 neurons, and we hope to increase into more than 1 millimeter. Of course, we also want to go deeper. You know that the 3-photon Miniscope is under development in other labs, and we're also interested to explore a little bit of that direction. And we also want to do multicolor imaging. So which means that we want to introduce multi-laser line excitation. So all of them are in our plans, and so the development is still going on.
Very thank you. So we probably can have a few more minutes of questions, and
I think I see some questions in the chat.
Yeah, we had one before, and I read it before, but
okay, yeah.
If there's any other question.
Oh yeah, sorry,
unmute.
Sorry to jump in. Yeah, I think I forgot to answer the last half question from Isabel, but dual channel. So currently, indeed, I mean, because the detector, you can see that the detectors are PMT, which is on the remote part. The detector is not integrated into the Miniscope, which means that we can record as many channels as you want. You just add more PMTs. And currently, the excitation is 1 wavelength, which is 920. And in 920, we can record some fluffers. For example, the GCAM we can image, and at the same time, because most of the times you only need 1 laser wavelength in 2-photon imaging to excite multi fluffers. So now we can also exact TD tomato, mRuby, or some RFP with the same laser, which means we can do the multi-channel. Yeah.
Good. So I had also a neurobiology question about some of the results you were showing, which I thought were incredibly interesting. So, but specifically, I'm curious about of the cells that you observe the activity in correlation with the behavior. So how many in those areas that you looked at, how many active cells do you find that are not grid cells or head direction cells or border cells?
Exactly, that's a good question. I think I have the data here. Okay, yeah. Sorry, I think there are some. It's here. If you just increase it, let me. Oh, this is not here. Anyway, yeah. So we found it really depends on where you implant your prisms, and it's highly variable. And in this, we call the hotspot, we found about 37% of the cells, they are gray cells in this particular region. And that fits also our observation from the neuron pixel recordings or in my colleague open host studies. And but the rest of the cells in these particular regions, they are really have direction cells or border cells, which means that the gray cells, they are really confined and they really occupy a specific terrestrial here. And the rest of the cells, some of them are similar to have some spatial tuning, but we cannot classify them to any classical spatial tuning cells like gray cell have direction cells. And in these areas, in this paraspeculum and also even other faraway regions, and the majority of cells, they have had the direction tunings. I forgot what's the percentage, but I believe that's higher than the gray cell ratio in MEC, but really less gray cell deaths. I think that will be the percentage in MEC. And the percentage of the spatial tuning in visual cortex, they are much less. They still have some, but much less. So yeah, that's the region. But it really depends on, sometimes we have animal that we hit not a very hot spot, we have 0 gray cells. And we still don't know why it happened.
Fascinating, yeah. We look forward to read more about this in some of the reports you're publishing. So finally, I had another question. I mean, you have shown here a very incredible new knowledge about observation of the activity, right? But in your description, you actually say there that you're working on new approaches for using all optical methods for functional connectivity characterization. Yeah. And so I was just really curious because you didn't mention any of that in the talk, and so I wonder if you're working now on that direction, and what are you doing in the direction of all optical mapping of functional connectivity?
Let me think. Let me check if I have. Yeah, because I wasted some time because time zone. I decided to just skip that part. But maybe I can just quickly show you some of the results. So the idea is that we want to measure whether the gray cells, because we are really curious about how these gray cells are generated, right? We know that the gray cell, they have this beautiful hex pattern of the grid fields, and some gray cells are really similar to each other, some are really different from each other. And the one theory of the origin of the gray cell is this continuously. Let's see if I have this. We call it the continuous attractor model. And a lot of you using this computation model to explain how the gray cells are generated. And to be brief, that this model assumed, or they provide a hypothesis that the gray cells, they are quite similar to each other. They should have really strong executory connectivity between each other. And the gray cells that are quite different from each other, they shouldn't have this strong, or they have really weak, or even the opposite, the inhibitory connection to each other. And we want to test if this is the case. And that's the motivation. And of course, with the Miniscope, we currently can only observe the activity, and we're not able to manipulate the activity. So then, in order to test this hypothesis, that is what I'm currently doing. Is that we want to using the optogenetics, we want to combine this Mini2P imaging to a benchtop two-photon stimulation and imaging systems, and which means that we will first use Mini2P to record, to detect these grid cells, and then we have another customized two-photon imaging and stimulation systems, and then in that step we have to have fixed animals, and then we try to align or register the field of view of Mini2P to the benchtop. So in this way, we found the exactly same grid cell in these benchtop systems, and we also express the option into the grid cells, and then we started to stimulate the cell one by one. And the idea is that if the cell they are connected to each other, and when we stimulate the presynaptic cells, we should see the response of the postsynaptic response. And if we see this response, which we know that they are connected to each other. And so we want to use this method to test the functional connectivity. And so that's the current goal. But this is really in a beginning stage. We haven't got any data yet, or very meaningful biology data yet. That's why I skipped this part. But I hope you understand the idea. Yeah.
Of course, very interesting topic for the future. And of course, if you want to also this stimulation and freely moving animals, there you have many other technical challenges for the future.
Yeah, we haven't have any plan. We have plan, but we haven't have any very clear idea on how we can also do this stimulation cell by cell in 3D moving in the Miniscope. Of course, we need fast stimulation. If you just want to stimulate one by one in a slow speed, it's possible, but we want to stimulate very fast, and we haven't started this project yet.
Very nice. So I think we have covered a lot of ground with the questions. So I just wanted to thank you again, Weijian, for dedicating this time, and we will send you the link to our talk online so you can share it with anyone who's interested also.
Yeah. Thank you so much.
Thank you very much.
Yeah.
And
okay.
Congratulations on all your work.
Thank you so much, and keep in touch.
Thank you. Bye-bye.
Bye-bye.